Hi, everybody. My name is Emma. I am a second year master's student at University of Minnesota. I'm also short. I work with Jess Goodconnect and Jake Youngers, and I mostly study soil changes in intermediate wheatgrass versus annual corn soy cropping systems. So today I'm going to introduce you to the experiment that I mainly work on and talk a little bit about what our goals are and what our initial results from this experiment are. So I work on this experiment called Perennial Annual Responses to Climate Extremes, or PARCE. It's located in Rosemount, Minnesota on a university research center. We're comparing 4 different cropping systems that represent a range of perenniality and soil disturbance. So I've got an intermediate wheatgrass and alfalfa intercrop, intermediate wheatgrass monocrop, a corn soy system that's strip tilled that has an annual cover crop, and a corn soy system that's tilled every year. This experiment was established, the first growing season was summer of 2020. And so we've had 2 growing seasons so far, and it will continue for 2 more growing seasons. As you can see in the picture on the left, I've got 20 replicates of each of these cropping systems. So we've got a good handle on the spatial variability of what's happening in the soil in this field. So generally the questions that we're addressing with this experiment are how do these 4 cropping systems representing differences in perenniality and soil disturbance affect soil health over a 4-year time span? So as you all are probably familiar with, we're interested in soil health and soil physical, chemical, and microbial properties because they can really affect crop yield and then also a lot of the environmental benefits or drawbacks of agriculture. So things like erosion, nitrate leaching, all of that sort of stuff. And we're expecting that growing a perennial system and growing using more sustainable methods for our annual systems are going to result in changes in the soil structure that really reduce the bad environmental effects of agriculture and also increase the climate resilience of yield in our agronomic systems. So as you're all aware, climate change is causing more frequent flooding and more frequent droughts, specifically in Minnesota. That's what we're getting, different across the world. But we're interested in how our 4 cropping systems respond to drought and flood scenarios. So to that end, starting this year, we've got some climate extreme treatments. So we're putting some of our subplots under these little structures to take 50% of the rainfall off of them through the growing season and simulate drought. And some of our subplots we're flooding with overhead sprinklers. We're adding 2 to 4 inches of rain every month to simulate that flood experiment. So the main project data categories that I'm working with, I'm not going to get into my methods because I think maybe a lot of people are not familiar with them. But if you want to talk more about that, come see my poster. I'm generally looking at soil microbial community composition and function. And then soil physical and chemical properties, both normal agronomic stuff, and then also some measurements that are commonly used in the soil health world to sort of quantify soil health, even though it's not a super quantifiable thing. And sort of a third research objective of this project is to explore the links between cropping system outcomes, soil microbial, physical, and chemical properties, see how they co-vary. So our results so far are not super interesting. We haven't seen any differences in the soil microbial community or soil chemical and physical properties from the first year of the experiment, which makes sense. Soil takes a while to change, and I'm working on my year 2 data sets now. So I just wanted to thank everybody who helps with this field experiment. It's a lot of work. Yep, and you can come talk to me at my poster if questions.
Hello, everybody. I'm Lydia Nicholson. This is my wonderful, beautiful colleague, Claire Weinman. And we're going to talk to you about the project we're working on called Kernza. All right. So basically, the first step is, what is this? So Kernza in Context is an educational curriculum that is designed to use Kernza and the research around it as a pathway to teach people about a variety of subjects, whether that be from social sciences to political sciences and regular normal sciences. We have focused on the second year on a high school audience because that's a level that should be accessible to adult learners who might want to use this curriculum to teach themselves, as well as being easy to edit to be usable in an undergraduate setting or for middle school age groups.
So we're conducting this project as part of the Kernza CAP, which you all heard about earlier, and it definitely reflects a lot of the collaborative nature of the CAP that Jake and Tara talked about. And so we get to work with a wonderful variety of collaborators on this project, including the EEP team, and then a group of fantastic graduate students who have been helping us to write some of these modules. And then we're especially excited because as part of choosing this high school student audience, we've been able to get to know a variety of high school teachers in the Salina area, and then put together a high school teacher advisory group of 3 teachers from Kansas and 3 teachers from Minnesota who have been working closely with us to evaluate this content we're creating to make sure it's highly usable and enjoyable in their classrooms. And so that graphic on the left is just kind of meant to illustrate that layered collaborative nature of the project.
Okay, so here we have just a list of the lessons that are currently in development that we're hoping to have finished by this fall so they can move into a beta testing period. Obviously, the point is not for you guys to read all of these lessons right now. If you're interested in seeing them, we have a pamphlet later that you can have access to actually read them. But the main point we want to talk about here is our framework and how we're thinking about these. So this is a modular curriculum, and these lessons are the smallest component of those modules. So each one we're hoping will be about a 50-minute class period that can be extended or maybe condensed a little bit. And then all of these lessons will be grouped together in larger chunks to form modules around certain subjects. So for example, Kernza and climate chains or Kernza supply chains. Some of these lessons will be individual to those modules. So I don't know, let's see. Kernza supply chains and systems will probably only be in that supply chain module, but other lessons can be used for multiple modules. For example, Kernza and water quality could be used in both a module about environmental qualities and things like that, but also a module just smaller communities and how Kernza affects agricultural communities.
And kind of as Lydia said earlier, obviously these lessons and modules are covering a wide variety of topics, and that was something that Aubrey and our previous post-bac collaborator, Megan, really discovered during the first year, doing a lot of interviews with teacher researchers and learning about how educators really want to see Kernza situated not only within the Kernza context, but also the broader community and agroecosystems. So looking forward just at the next couple of months of the project, we're going to continue with this lesson development and revision, hoping that by midsummer we'll be able to make these modules and lessons publicly available for different teacher researchers to actually beta test in their classrooms as part of this beta testing process that we've developed. And we especially wanted to highlight that for you all in case any of you might be interested in participating in that beta testing process. And so we'd love to either be contacted by you all by email or discuss here at the Kernza meeting to make sure that we can get you on that list to receive those modules. And then this afternoon, we're going to be helping our wonderful collaborator, Hannah Stoll, host a breakout group where you're actually going to get to engage with one of the activities that's in one of these Kernza in context lessons. So if you'd be interested in experiencing that yourself, you should absolutely come see us. So we're thrilled to be part of the Kernza CAP, and we're thrilled to thank you.
Yeah, thank you.
We know that there are environmental factors that influence disease resistance in the field, but there's also a genetic side of that. And as we heard earlier this morning in other talks, genome-wide association studies are a way that we can identify genomic regions that are contributing to that disease resistance. So the purpose of this project was to identify some of those loci that may be contributing to common field pathogens such as Fusarium head blight, bacterial leaf streak,
and ergot. So we used the C7 breeding population, which was the first to be used for genomic selection, and they were space planted here in Salina, Kansas. And each year, 2019 and 2020, they were inoculated with Fusarium head blight infected maize kernels, irrigated, and then we evaluated them in the field and after harvest for the list of 18 traits seen in the table on the slide. Then after harvest, we also sent grain samples to North Dakota State University, where they used gas chromatography to measure the deoxynitrolenol or DON content and also its detoxification product, which is DON 3-O-glucoside. As I said, these plants were genotyped for genomic selection, so we had genotype by sequencing markers available to us, and I used the farm CPU function of GAPIT for the GWAS to assess the marker trait associations. What we found in the field was that many of our Kernza plants did have some disease resistance, even after intentionally inoculating with the disease. And like we like to joke about at the Land Institute, our program intentionally tries to make the plant sick. So even with that, we saw low disease levels. And overall in the GWAS, we saw 333 significant loci for 15 of the 18 traits. 0 loci were found for Fusarium head blight incidence, grains per spike, or length of spike. And Fusarium head blight index was the only one that had 1 significant loci, and all the other 15 traits had more than that for significant loci. The most were found on chromosome J2 and J7. And unfortunately, there were not any loci that were the same significance every year. However, there were 2 for Fusarium head blight DON content that were significant between a few years. One was significant in 2019, 2020, and in an average of all 3 years. And one was significant for 2019 and 2020. And since DON is very inhibitive to the use of Kernza grain for humans, it's one that we want to look at more for disease resistance. And this trait was the measure of the content. So when we see this negative allelic effect, we conclude that it's contributing to disease resistance. I also included the percent variation explained and note that it is very low. But again, as we heard earlier, that's seems to be common with a lot of these loci, and showing that there's a lot that are contributing to the overall resistance of the plants. And I also included one of the Manhattan plots that shows these 2 significant loci from this one specifically from 2020. So we're seeing other loci are also significant, but I pointed out the 2 that were significant in several years. So to conclude, the identification of these markers for DON and other traits can help us in future breeding cycles and hopefully improve the disease resistance of plants. These are acknowledgements to the funding of this project, and also thank you very much to Catherine Turner at the Land Institute and Jared Crane, who have helped a lot with this project and in my own education as a postbac.
Hello everyone, I'm Anusa Dahal and I'm the first year master's student in Department of Plant Pathology at Kansas State University, and I'm working as a graduate research assistant in Dr. Jessica Robb's Applied Wheat Pathology Lab, and I'm working on the Kernza project. So today I'm going to walk you through the methods that I have been doing in our lab. So we received 70 grain samples mainly from Kansas, North Dakota, and Minnesota. And so I processed the samples by first by starting with distillation and then cultured the samples. And after several soft culture, I was able to obtain this mycelium in the plate, and we culture the samples in SNA media. So after obtaining this mycelium, they were again cultured in the mung bean broth for the production of macroconidia, and these macroconidia were diluted, were counted in the hemocytometer, and then it was diluted for the production of single spore. And I used this single spore for generating the single spore sample, and this sample was used for extracting the DNA. So I used CTAB extraction protocol for DNA extraction, and similarly I used the ITS for doing the PCR of these samples, and the samples were sent for sequencing. So far, I have been able to do the sequencing of 40 samples, and from that, what we have found that 32 samples are identified as the Fusarium. Yes, and we came in the conclusion that it was well known that Kernza is kind of resistant immune crop, but I doubt that after this result, we can't say that Kernza is resistant crop. So we need to do more research on this. So I have this question here that we are well known about the Fusarium head blight infection in the wheat crops. So now we have identified the Fusarium species in the Kernza as well. So I think it would be problematic with the shared Fusarium species in wheat and Kernza and many other crops. And we had also received the leaf samples, and while we do the nanopore sequencing and whole genome sequencing of these samples, we found out that there were several viral disease and the leaf spot disease present in the Kernza leaf species. So there might be other endophytes and other pathogen as well present in the Kernza crops. So we might be doing some kind of unseen loss in our crops. So I think we should also focus on this area as well. So the future work in our lab will be after identifying all the samples with Fusarium, we will be performing the PCR by Fusarium specific primers to go into the species level, and then screening of wheat as well as Kernza will also be done, and we'll also calculate the disease infection percentage and we'll evaluate the disease for obtaining our objectives of selecting the resistant varieties of Kernza. So if we have time, I will take the questions later. So thank you everyone for hearing me out, and I would like to thank our funding source, North Central SHARE, and for providing us with the funding, and the Land Institute for helping us by providing samples, and our supporting partners, Kansas Wheat, USDA. So thank you.